Signal transduction pathways. A signal transduction pathway is a process in which we take an outside message, or a hormone, for example, and creates a series of events inside the cell and have chemical effects. It all starts off with the primary messenger, or the outside message, which in this case will be the ligand. The ligand attaches to a very specific spot on the receptor protein. In this case will be the G protein. The G protein has a very specific spot which only the ligand can attach to. The ligand makes the G protein loose off an alpha unit. The alpha unit that was broken off from the G protein moves over to the adenylyl cyclase. The alpha unit causes the adenylyl cyclase to make ATP. The newly formed ATP will lose 2 phosphate bonds, thus creating cyclic AMP. The newly created CAMP only has 1 phosphate group, which is why we call it cyclic monophosphate. As we move further into the cell, we can see a protein kinase. This protein kinase is made up of a catalytic and regulatory portion. The cyclic AMP or CAMP bind onto the regulatory portion of the protein kinase, thus freeing up the catalytic portion of it. The freed up catalytic portion of the protein kinase will receive phosphate groups. This process is called the phosphorylation cascade, as it occurs on a grand scale. The catalytic portion of the protein kinase, now activated with the phosphate groups, or charged up with the phosphate groups, will transfer the phosphate bonds or groups onto the phosphorylase. Then the phosphorylase gets activated and starts breaking down the glycogen into monomers called glucose.